Hello everyone, welcome back to RPIT Academics. I am AVIKA, working as an assistant professor in RPIT Technical and Medical Campus, Bastada, Karnal. Today we are going to discuss about the pharmaceutical chemistry. So, first of all, we will look for the scope and objectives of pharmaceutical chemistry. What is pharmaceutical chemistry? It is that branch of chemistry which deals with the study of organic chemistry, that is molecules and compounds which contains carbon atom in combination with structural and chemical biology and pharmacology for producing pharmaceutical drugs and medicines. So, in pharmaceutical chemistry, it comprises of drug development and the study of drugs, which includes drug discovery, metabolism, absorption, and delivery. They contain drug chemistry, quality assurance, metabolism, pharmacology, and analytical techniques in cures and remedies disease. Now, if we look for the scope of pharmaceutical chemistry, it in quality assurance and quality control, that is QA and QC, processes and standards that ensures quality of drug compound. In drug discovery, which is identifying compounds, especially those that treat disease, in it is used also in industries and pharmaceutical chemistry teacher for college institute, etc. Now if we look for the errors, what errors are? Errors is the mainly we can say error, this is the difference between the true value and the measured value. It is defined as deformity present in any measurement by addition of any internal or external factor. In pharmaceutical science, errors are induced by the defective equipments and methods. In analytical chemistry, errors are effects that material produces reliability, reproducibility, and accuracy or precision. During an analysis, the results are expected to be highly accurate and precise. However, it is not happening in all cases because due to the presence of errors. Now, errors may be predictable or unpredictable, both. That depending on the calculative nature, errors can be categorized in two parts. That first one is absolute error and the second one is relative error. Absolute error means it is the difference between experimental mean value and the true value or actual value, which is already known as absolute error. Absolute error may be positive and negative both. We can calculate the absolute error by subtracting the true value from the measured mean value of the experiment. Now, if you talk about the relative error, it is defined by dividing the absolute error by the true value. It is generally expressed as percentage. This is by the multiplying the relative error by 100 or by expressing it as parts per thousand by multiplying the relative error by 1000. So, on the basis of nature and sources, errors can be categorized in two types or two principles. First one is determinate, which is also known as systemic error, and the second one is indeterminate, which is also known as random error. So, first of all, we will talk about the determinate or systemic error. This error can arise due to the wrong procedure, wrong measurement, or faulty instruments, that is uncalibrated balances and machinery system. So systemic error is under the control of the analyst because it is easily detectable and can be eliminated to a large extent. Now, what can be the sources of systemic error? These may be instrumental error, means due to the use of defective or low quality instruments. Proportional error, proportional means absolute value of this kind of error changes with the size of sample. If the size of sample increases, then the value of the error also increases. Next is personal error. It is induced due to the carelessness, ignorance, and lack of skills, or untrained analyst. Next is chemical or reagent error. This error based on the chemical reactivity between using chemicals and reagents. Next is error in methodology. The method or the procedure which is employed by the analyst is responsible for such type of error. Next, second one is indeterminate or random error. In this error, we do not define the specific well-known reason and cannot be eliminated. So it is also called as accidental error or random error. It is induced by several successive measurements performed by the same analyst under the same conditions and identical experiments. Thank you and have a nice day. If you like the video, please share and subscribe our channel.